This table summarizes the extracellular signals, their source, and effects at various phases of wound repair. It provides a snapshot of the key molecular players that are orchestrating the dynamic healing process, such as coagulation, inflammation, granulation tissue formation, angiogenesis, contraction, reepithelialization, maturation, fibroplasia, and arrest of proliferation, and resolution and remodeling. So, these are the various phases of wound repair. First is the coagulation that needs factors 13A, TGF alpha, TGF beta, platelet derived growth factor, ECGF, and FGF or fibroblast growth factor. Their sources are plasma and platelet, and they are responsible for cross-linking of fibrin thrombus that strengthen the clot, and they also act as chemoattractant of other cell for the subsequent healing phase. Then there is phase of inflammation. Here the factors needed are transforming growth factor beta, chemokines such as TNF alpha, interleukin 1, interleukin 6, CXCL12, CX3CL1, and platelet derived growth factor. Their sources are neutrophils, M1 macrophages, and endothelial cells, and the effect produced are recruitment of monocytes and fibroblasts, and activation and differentiation of fibroblast and stem cells. Then there is granulation tissue formation. The factors required are FGF2, transforming growth factor beta, and hepatocyte growth factor, and the sources are keratinocytes, monocytes, and fibroblasts that affect as various growth factors. They are bound to proteoglycan matrix and they stimulate fibroblast proliferation and migration. Then there is angiogenesis, that means new blood vessel formation, and the required factors are VEGF, FGF, hepatocyte growth factor, angiopoietin 1, 2, and PDGF. The sources for these factors are monocyte, macrophage, fibroblast, and endothelial cell, and the effect on blood vessel growth and also on pericyte to stabilize the new blood vessel formation. Then another step is contraction. Here factors required are TGF beta 1 and beta 2, and these are derived from macrophages, fibroblasts, and keratinocytes. The effects are myofibroblast differentiation, binding to the collagen, and they also generate tension leading to wound contraction. Then there is reepithelialization, and the factors required are keratinocyte growth factor or FGF7 and hepatocyte growth factor, epidermal growth factor, HB, TGF, TGF alpha, activin, TGF beta 3, CXCL10 and 11, and their sources are macrophages, platelet, fibroblast, keratinocyte, and endothelial cell. They affect as proliferation on the keratinocyte. Also, they exert their effect on the migration and restoration of the barrier function. The next step is maturation, fibroplasia, and arrest of proliferation. Here, the required factors are TGF beta 1, platelet derived growth factor, CTGF, interleukin 27, 4, and CX3, CL1, and thrombospondin. The sources for these factors are M2 macrophages, fibroblast, and keratinocyte, and they affect as they are responsible for extracellular matrix accumulation, fibrosis, and tensile strength. Then there are additional extracellular matrix proteins such as heparan sulfates, proteoglycan. They capture TGF-beta, VEGF, and basic fibroblast growth factor in the basement membrane. Then there is decorin that captures TGF-beta, stabilizes collagen, and downregulate fibroblast proliferation. Immune modulation also, it requires interferon CXCL10, 11, and it suppresses fibroblast proliferation and accumulation of the collagen. There is increased oxygen decrease mechanotransduction that support maturation and reduce fibroblast proliferation. Finally, there is a resolution and remodeling that requires PDGF, FGF, TGF-beta, interleukins, MMP, tissue plasminogen activator, urokinase plasminogenic activator, and tissue inhibitors of metalloproteinases, and these are derived from platelets and fibroblasts, keratinocyte, macrophages, and endothelial cells. Their effect is on extracellular matrix remodeling. MMPs and TEMP, they regulate collagen turnover, for example, replacing collagen 3 with collagen 1, and they are also responsible for balance of MMP activity to ensure proper restructuring of the extracellular matrix. Then reduced cell density limit excessive scarring that is caused by CXCR3 signals. Final tissue remodeling stabilizes the tissue structure and ensure long-term integrity. So each phase has a specific signal source and tightly controlled effect. The MMPs and TIMPs, they play pivotal roles in the later stages of extracellular matrix remodeling, while earlier phases focus on inflammation, recruitment of the cells, and matrix formation. The late phase involve deposition of extracellular matrix, their maturation, and final shaping of the scar tissue. Thank you for watching this video. If you have any query, please write in the comment box.